In a commercial food processing facility, discovering a single insect in a finished product triggers an immediate operational crisis. Entire production lines halt, and thousands of pounds of product are instantly flagged for potential destruction. The problem compounds as that localized finding implies a much larger threat. Through massive industrial distribution networks, a single point of failure can contaminate shipping containers, storage silos, and downstream retail partners within days. Standard generalized pest control treatments like broad-spectrum fogging or spraying are highly ineffective in these large-scale environments. They treat the symptom, not the source, leaving the hitting breeding population completely intact. Quality assurance auditors require exact data to authorize interventions. Identifying a discovered pest merely by its family or genus provides zero actionable intelligence for isolating the contaminated zone. Exact species-level identification is the only way to neutralize an infestation at its biological root. To achieve this, auditors rely on a foundational rule of structural pest management. Morphology reveals ecology. An insect's physical structure isn't random. It dictates its feeding behavior, its reproductive cycle, and its required target environment. An insect's microanatomy acts as a reliable map, allowing professionals to trace the infestation backward through the facility to its exact commercial food source. Navigating this requires a specific diagnostic matrix, one designed to differentiate pests that appear nearly identical to the untrained eye. This diagram isolates key microanatomical zones. The largest diagnostic marker is the elytra, the hardened forewings marked in red. Surface texture here splits lookalike species. Secondary markers include the yellow pronotum and antenna shapes. Mastering these allows instant ecological categorization. The first major category to identify are the internal feeders. These are pests whose larvae develop entirely hidden within a single, intact kernel or seed. The primary and most destructive internal grain feeders are true weevils, belonging to the family Curculinidae. True weevils are defined by two distinct diagnostic features: clubbed antennae and an elongated snout called a proboscis, which they use to bore directly into the center of hard grains. This scale comparison illustrates a common diagnostic challenge, distinguishing the rice weevil on the left from the granary weevil on the right. The rice weevil presents with 4 lighter X-shaped markings across its elytra. Examining the thorax reveals dense, closely spaced oval pits. Ecologically, rice weevils possess fully developed hindwings and are strong flyers. This drastically increases their cross-contamination footprint, spreading rapidly between distant storage silos. Contrast this with the granary weevil. Its elytra lack the light markings, and the pits on its thorax are distinctly widely spaced. Because it lacks functional hindwings, a granary weevil infestation is usually confined to the immediate grain mass where it was introduced. There is a notable exception within internal feeders, the bean and pea weevils. As seen in this lateral profile, they have a heavy teardrop-shaped body and completely lack the elongated proboscis of a true weevil. Despite this morphological difference, legume weevils maintain identical internal feeding behaviors, developing entirely inside individual beans and peas. Ultimately, identifying the distinct markers of an internal feeder dictates an immediate targeted inspection of the whole grain supply chain, ruling out any processed or milled goods. Internal feeders are not exclusively beetles. Specific members of the Lepidoptera order, moths, must also be accounted for in whole grain audits. The Anglemoa grain moth is the primary internal threat in this category, infesting corn, wheat, and barley. Adults are identified by pale yellow forewings that terminate in a highly distinct fringed edge, separating them from other common storage moths. This cutaway view shows the larva developing inside the kernel. Recognizing mechanically hollowed-out corn points directly to an internal feeder's early development, allowing auditors to spot an infestation even when no adult moths are present. The second ecological category are external feeders. These scavengers develop entirely outside the whole kernel, targeting processed flours, milled goods, and dried organic matter. Within this group, auditors face the two most commonly confused pests in the industry, the drugstore beetle and the cigarette beetle. Misidentifying these two threats leads to catastrophic inspection failures, because while they look identical, they target completely different processing zones within a facility. Both beetles share an identical oval shape. The elytra provide the first separation. The left drugstore beetle features distinct longitudinal grooves along its wing covers. This contrasts with the right cigarette beetle, possessing completely smooth, ungrooved elytra covered in fine hairs. If wing covers are damaged, professionals use a microscopic failsafe, the antennal structure. The cigarette beetle possesses highly serrated, saw-toothed antennal segments, labeled here as C. The drugstore beetle utilizes a completely different structure, terminating in a loose, three-segmented club, labeled as D. Distinguishing smooth versus striated elytra instantly shifts inspection parameters. A drugstore beetle points toward pharmaceuticals and baked goods, while a cigarette beetle redirects the search toward processed tobacco and dried botanicals. The final major category encompasses the family Dermestidae, which operate as advanced scavengers. Adult dermestids share a common macromarker, a stout oval shape with short clubbed antennae and a head highly retracted beneath the pronotum. As seen in this hide beetle, identifying the adult dermestid is secondary. The larval stage is far more critical for diagnosis. A dermestid larva is immediately flagged by its dense covering of stiff hairs, or seta, across a banded, cigar-shaped body. In species like the warehouse beetle, these hairs are specialized into spear-headed structures called hasida. This creates a severe medical hazard. If these specialized larval hairs shed into processed food and are ingested by consumers, they embed in the gastrointestinal tract, causing severe irritation and distress. Other dermestids, like the hide beetle, shift their ecological focus primarily to animal proteins, feeding on dried meats, pet foods, and hides. You can visually verify a hide beetle larva by checking the rear of the abdomen for two sharp, curved terminal horns, known as urogompi. Discovering these hairy larvae dictates the immediate quarantine of high-protein goods. Their presence introduces a direct consumer health hazard into the inventory. Precise taxonomy identifies the specific biological vulnerabilities required for successful facility management and targeted pest eradication. We can map these physiological traits onto a final sorting matrix based on two vectors: feeding behavior and target protein type. Pests equipped with a proboscis, or grain moths capable of hollowing out kernels, lock into the internal plant protein quadrant. The external scavengers, our striated drugstore and smooth cigarette beetles, map directly into the external plant protein quadrant. And the heavily haired dermestid larvae straddle the scavenger line, feeding aggressively on both processed plant goods and dried animal proteins. In the field, this framework dictates the immediate response. Finding a proboscis demands a full, localized audit of the whole grain supply chain. Finding smooth elytra and serrated antennae means widening the search perimeter into processed spices and flours. And discovering Hasticeta instantly flags an urgent recall hazard. This image of heavily damaged dog treats proves the physical result of an unchecked infestation. Mastering microanatomy is the only way auditors can read mechanical damage like this and definitively trace it back to the biological source of the collapse.